Continuing, we'll study about how cell cycle control system works. For this to understand, first we need to get what happened when during the cell fusion experiments. Okay, at that time, what happened was two living cells are they are fused at different times to form one cell with two nuclei with two nuclei. For first case, for example, if a cell was in S phase, okay, it is fused with G1 cell. Then what happened here? At G1 nucleus is triggered to start DNA replication immediately. Start immediately. Now second case was if a cell was in M phase, okay, then if it fused with G1 cell, or we can say any other phase, the nucleus showed the second nucleus showed some mitotic changes, such as chromatin condenses, spindle formation. These were the result of those two experiments. So basically, our question is how it was done. How it was done. To answer this, there is a system called cell cycle control system. Control system. Okay, it is driven by a molecular control system, which is, we can say, it acts at a time program, like it acts in a certain events in order, and then coordinates them to do the DNA replication, mitosis, cell division correctly. You at perfect steps. Molecular control system. Like there are some checkpoints and some signals to do this and do this process. This is maintained by this process. It triggers the event in cell cycle, and sometimes thing called, we can say it as checkpoint. I'll tell you about earlier. You can say it is a control point where stop and go signals can be found, can be done to continue cycle. So basically what just happened, in a cell cycle clock, you can say cell cycle is controlled by, for example, it is controlled by some molecules that work like a clock. We can say, the events like DNA replication, we can say, or division, it should happen at a right time. Okay, so these are this happens because the cell cycle use certain molecules whose levels are rise and fall, taking the clock. you know when the changes in molecules happens, the cell know to move to the next stage. For this to happen, there are two types of main molecules. First is, we can say, protein kinesis. It is an enzyme. Okay, the job to do, turn on or off proteins. They do this by phosphorylating or phosphorylation, which means adding a phosphate group. No, second thing is, say, cyclings. These are the main things, you know, like whose amounts go up and down during a cycle. Okay. So they are concentration cycles of concentration. That's why they are known as cyclins. Like in cell, most of the kinases are present. This is present, but like they are inactive, not active. So they need something to do work for it. For the something is cyclins. Why? Because this pair forms kinase plus cyclin. It results in active. Okay, and this all thing is called as CDK, which means cyclin dependent kinases, which means CDKS. So it is directly proportional, like when cyclin level rise. Okay, more CDK activity. It is directly proportional. We can say cyclins directly proportional to kinase to induce more activity. Okay, if cyclin level falls, you know CDK activity stops. So now, if for example, the first discovered cyclin dependent kinase complex, discovered, it is found in frog eggs. Now, there's one thing called as MPF, which is known as maturation promoting factor. Okay, now the MPF, this activity peaks when the cyclin rate is up. When the cyclins are producing more and more, concentration of cycling is highest, this activity please. Now in the cell cycle, in S and G2 phases, the cyclin accumulates highest. It's more, we can say highest. Okay, so MPF becomes increasingly active, more active, you can say, and this helps to trigger the cell into move to M phase, which is, you can say, mitosis. In cell cycle, S phase and G2 phase, cyclins rise. Okay, start of meiosis, this leads to start of mitosis. Okay, cyclin plus CDK, you can say high MPF activity. End of meiosis, you can say M phase, mitosis. Cyclin is destroyed. Okay, causing MPF activity drop suddenly. This helps cell cycle to finish and return to G1 again. Now MPF is like, earlier it is known as M phase promoting factor. but now it is converted or like change its name to maturation promoting factor, because like it is the main step to induce from G2 phase to M phase. Now like the cell, like MPF pushes the cell to past G2 checkpoint. If MPF becomes active, it passes G2 and active passes G2 and enters into M phase, which is we can say yes signal for mitosis. Now how it becomes active? Okay, like there are two components I told you earlier, cyclin and CDK, cyclin dependent kinases, cyclin dependent kinases. It builds up during S and G2 phase. Okay, it is always present but inactive. Now what happens is like when cyclin accumulates, okay, wait, when enough cyclin accumulates in G2, okay, it binds to CDK plus cyclin. They form an active MPF complex. Now what does MPF do later on? It adds, we can say, phosphate groups, which is known as phosphorylation to many proteins and which induce mitosis. Okay, it acts in two ways. Like first we can say as kinase, like it start like phosphorylation on protein itself, or second one is we can say indirectly by activating other kinases. This induces a chain reaction. Now what happened in cell, if like MPF it phosphorylates proteins in the nuclear lamina. Okay, more and more proteins are there. When these are phosphorylated, lamina, we can say, it breaks apart inside the nucleus. For example, this is a nucleus. Okay, wait, this is a nucleus. Okay, inside these are MPF, that's inducing more proteins and proteins. This is a nucleus. We can say this is cell. This is a nucleus wall. Okay, like it's a membrane of nucleus. We can say. Okay, when the nuclear lamina breaks, okay, it breaks the membrane also. It is done by adding more proteins by adding a phosphate group. Like this process happens in the phase of, we can say, pro-metaphase. Secondly, what does this do? Like MPF also activates the protein, we can say, that tighten chromatin tightens and condense. You can say chromosomes get condensed here also. Condense. This happens in prophase. Furthermore, spindle forms. at metaphasic plate. Now during the anaphase, this is important thing to note, MPF, you know, destroys the cyclin. When the cyclin is destroyed, MPF becomes inactive. Okay, so mitosis move toward completion. This was the end of the M phase. Now, important thing is like CDK still present in the cell, but it is inactivated. You know because like cyclins are not present. Now later at the new cell, like when those two cells divided, at that stage when the S and G2 phase forms, the cyclin will rise again. Now some general things like MPF is not only the one like we initiate like this to induce the mitosis. Okay, it's not only cycling CDK complex. There are other one also. There are many multiple types. For example, we can say like MPF. It controls G2 and M phase transition happens there. Other cyclin CDK complex, cyclin complex. It controls, you can say, G1 checkpoint. Another one we can say controls G1 and which leads to S transition, S phase. Each complex, you can say, acts as green lights, which give the green signal to induce next part. In animal cells, there are at least 3 CDKs, okay, and several, you can say, G1 cyclins. These work together to pass the G1 checkpoint in animal cells. If at G1 checkpoint, if the cell does not goes further, it goes into phase we can so called as G0, you can say resting phase. If the cell repairs or like some damage was happens and the problem happens, it repairs here and then again it starts again. For example, at G1, many C and cyclin CDK complex check the cell can divide or not, further or not. At G2 checkpoint, which like transitions to M phase, here MPF give the final go signal. At M phase, MPF starts chromosome condensation of chromosome, you can say spindle formation and breakdown of nuclear envelope. At the time of anaphase, MPF destroys cyclin, so resulting is turning off.